Thank you, organizers, for giving me the opportunity to talk about what we are doing in Noble Foundation. So today I'm going to talk about a unique fungi and how it promotes growth. And so basically our model is to promote sustainable agriculture using of low input or no input fertilizer and improving plant health and plant fitness. So in first, why microbe in first place? So we all know that in our body we have hundreds and thousands of microbes, and they actually help us in many ways. They help us to remain healthy, and which most of the times we are not aware of. Similarly, like our body have our own microbes that helps us to keep healthy, plants have their own microbes that helps the plant to keep healthy. And more we rely on like chemical fertilizers, chemical inputs. We kind of lose these plants' natural microbes, and as we more and more depend on medicines, we kind of compromise with our natural immunity, natural defense systems to fight diseases. Similarly, plants lose these microbes with the over usage of chemicals, whether it is a fertilizer or pesticides or any forms. So with this, I begin about these unique fungi. And so these unique fungi are a root-associated fungi, and which has the ability to improve plant growth. And some of you are aware that this type of associations we call as mycorrhiza, and these mycorrhiza are two words joined together, in which mycos means fungi and rhiza means root. So basically these are root-associated fungi, and they helps in plant growth. So this particular fungi which I am going to talk about is unique in many aspects. And one aspect is that these fungi are found kind of almost everywhere. So far we have not found a place where this fungi does not exist, and they have a very wide spectrum of association with plants. So these fungi can make association with lower forms of plants, higher forms of plants, and even brassicasses, like, which is traditionally known as non-mycorrhizal. So thus far we have not found any plant which has shown any kind of resistance to this group of fungi. So we have tested these fungi with various types of crops, and I put here 3 examples of monocots and 3 examples of dicots, where we have seen that in greenhouse conditions and controlled plot experiments, we have found that these fungi, along with plants, improve plant biomass in a variety of levels. So how a single fungi is able to colonize such a wide array of crops? To understand that, I present this small cartoon. So basically, if you look at this picture, so this is a cross-section of a root, and this picture actually tells about how different types of mycorrhizal fungi makes association with different types of plants. So these are different ways. Okay, so these are actually fungi. These are the filaments of fungi which grows out of the roots and fetches nutrients from the soil, and they can form this type of hair-like structure. They can form this type of spores, and inside the plant's roots they form this type of structures where all the nutrient exchanges takes place. And this can be of this type of hair-like structure and this type of dense packagings. So basically they collect nutrients from outside the roots, and they transfer these nutrients inside the roots, and then they move along the entire plant. This is how actually looks like in real situation. So all these green colored things which you see, these are actually fungal structures heavily packed inside a single root cells. So this is tall fescue, this is switchgrass, and this is winter wheat. And this is a single root cell, and this is how they are packed inside. Basically, this technique is called confocal microscopy. And what we do is we take the picture of fungus with a green filter, then we take the picture of a plant root with a red filter, and we combine them together, and this is how it looks like a plant root which is packed with fungi. So basically this is the place where all the good things happen. So as you have heard from previous speakers, so fungi cannot synthesize their own carbon because they are achlorophyllous. They do not have the ability to produce their carbon, while plants are photosynthetic plants, so they basically synthesize carbon using the sunlight. Now, why would such relationship would happen in spite of the fact that sometimes, why would a plant will allow a fungi to live inside its own body? And the reason is that both are actually getting little bit of benefit. So in case of fungi, they are getting a carbon from the plants. And in case of plants, they are getting all the macro and micronutrients from the fungi. However, this is actually a cost-effective thing for the plants, which Keith said in his talk that this is not free. Like plants have to pay a currency to maintain these fungi within their body. And if you add too much of fertilizer, then plants do not allow or do not need these fungi to live within their body, because it's simply put like fertilizer are some kind of free food to these plants. So fungi, when the plants get these free foods, they really don't want to work hard, and they don't want any kind of this type of friends. But when you keep the fertilizer dose to a limited amount or low or no fertilizer, then they actually look out for these friends which actually can go out into the soil and bring all these good nutrients and give it back to the plants. And in long term, this is actually win-win situation for everybody, because over the years, if you keep on adding fertilizer to the soil, you actually compromise with the soil health. More and more fertilizer you add, you compromise with the diversity of microorganisms present in the soil. You compromise with the soil's porosity, and there is no end to it. On the contrary, if you rely on this type of good and beneficial microbes, it's kind of you are using nature's own method to keep these plants healthy, to keep our soil healthy. So with this, I talk about a case study where we have used winter wheat, and we have tested how this fungi, which helps plants to grow better and in field conditions. So we all know these things, like why wheat is important. Wheat provides like 20% of the all calories consumed by the people worldwide, and world's population is ever increasing. And basically, if we want to keep the cost of production low, we need to have a sustainable method where we can feed this ever increasing population in a more sustainable manner. So basically, we need a method which we can sustain over years. I'm not talking of 5 years, 10 years. I'm talking of much more beyond that. Now, I have told you about these beautiful fungi which can provide a lot of benefits to the plants. Now, we need to have a method how we can use these fungi to go to the field and inoculate crops in the field. Now, there are a lot of methods available to do that. So, here we tested different types of methods to how to deliver these fungi into the soil. One method we tried is we have used wheat straw, and we put fungi in this wheat straw and see if it grows. We choose wheat straw because it is cheap and it is readily available, it is easy to manipulate. And another method which we used, it is called root organ culture. What we do in this method, actually, we grow only the roots. We call this as transformed roots. Basically, these roots without any shoot, without any, they can grow in the laboratory conditions. And in this method, what we add these fungi, these green thing, these filamentous cottony things, we add these fungi into these roots, and we grow them in the laboratory conditions, and then we put it back into the field. Another method is like growing little plants in a controlled environment and adding fungi to the roots. And finally, about which I'm going to talk about today, using these clay particles. These are basically what you commonly call as starface. So use these starface and add fungi to the surface of the starface and put the starface into the soil to deliver the fungi. So out of all these 4 methods which we have tested, we found the last one, the starface coated with fungi, as the most efficient one. And I will spend little more about how we actually use this technology for mass production of these fungi for field trials. So what we do, actually, we grow fungi in bottles along with those clay particles, the picture which you have seen just in the previous slide. And once these particles are completely coated with fungi, we take them out and put it in some synthetic media in the laboratory conditions, and we see the fungi are actually coming out. So this is an indication that these small clay particles are actually coated with fungi and they are still alive. So this is the same individual clay particle with a bigger size. So this is a clay particle which does not have a fungal coating on its surface, and this is a clay particle which have this fungal coating on its surface. So these white cottony things which you see, these are actually fungi coated on the surface. And when we look deep down under an electron microscopy, where we magnify it by 1,000 times, we can see these small hairy things present all over on the surface. Now, so far in this, we know that these clay particles can be coated with a fungi, and in this coating they are still alive and they can grow again out of these particles. But how we use this thing to actually infect a plant? So what we do in our lab is, actually we use these small tubes, and we fill up these small tubes with these tiny particles, and each of these particles are coated with fungi. Then we grow a plant inside this, and as this root system goes down, they actually comes in contact with this each and every individual clay particle, and fungi from this clay particle enters the root system. And this is how the fungi from the clay particle can enter the root and successfully colonize the plants. So this is another picture for a better idea. So this is how these clay particles are covering, these fungi are covering these clay particles, This is how you see in the field conditions. These filamentous mycelia are coming out of these clay particles. And another picture where you see this green in the sea, the green bluish thing, which is actually the fungi. So far we are able to show that these clay particles can be successfully coated with fungi, and in this model these fungi are viable and they can also infect successfully colonize plants. But we wanted to know that how is this better than other method of delivery currently available in the market. So what we did actually, we actually tested if this fungi plus clay particle combination actually releases macro and micronutrients which can further improve plants' growth. So what we did actually, we took these clay particles coated with and without fungi, we just put it in the water and shake it vigorously, and took that water and analyzed for the various macro and micronutrients. And so if you look at this, so I want you just to concentrate on this one. So basically, because of the presence of the fungi, all the good nutrients are more released into the soil, and these are fungi actually consumes it, so they are not released. So this is with respect to the control. You can see how the nutrients is more and less. So simply put, that we are able to demonstrate that these fungi in association with the tarface can actually release micro and micronutrients which will be helpful for the plants to grow. So this is an example how these fungi increases the plant health. So this is wheat, winter wheat in these small tubes which I have shown you before. And look at the root length in presence of fungi and in absence of fungi. So this is how this unique fungi helps. They actually accelerates the plant's growth in a much shorter time. So they produces bigger roots which helps in greater nutrition acquisition, greater water use efficiency, and this is how it helps the plants to grow. So now when you took this small piece of root and look under the microscope, you can see all those green things. So these are all fungi present all along these roots, and this is just a much closer look of these fungi, how they are present inside. So far we were able to successfully develop a method where we can coat this unique fungi in a medium and which can successfully infect plants of our choice. Now our next goal is we wanted to know that if this actually helps across the 4 different cultivars of a plant. So for this experiment, we choose wheat as our choice of crop. We choose wheat because in Noble Foundation, wheat is one of the 4 important pillar crops we are currently investigating. So what we did actually in laboratory conditions, we grow 3 different cultivars of wheat. We got these cultivars from OSU, and this is control, meaning that these plants are without fungi and these sets are with fungi. So you can clearly see the difference between the presence and absence of fungi and in 3 different cultivars. So thus far we were able to convince ourselves that these fungi can improve a biomass of wheat under laboratory conditions. But our next step is that we wanted to know that if it is really true in field conditions. For that, we did an experimental design. We went out to the field and we did this for 2 years. So we choose 2 field locations here in Ardmore. One field location, we call it as NF1, which is an NF2. They are all in and around Ardmore. And we choose 3 different cultivars of wheat, NFEX25, Duster, and NFEX28, and 3 treatment with fungi. One is Sebacina, which means that we treated these plants with fungi. Control, in which we treated these plants with these clay particles without containing any clay particle. And the third one is absolute control, where we give nothing to the plants. So we run this experiment for 2 years in 2 different field conditions. So I just want you to concentrate on these green bars. Green bars are those plants which are treated with fungi. Yellow bars are those where these plants are treated with clay particle without any fungi, and the red bars are those where absolutely no treatment was given. So what we found is that for this cultivar NFEX25, we see an enormous enhanced in grain yield with respect to treatment, and whereas in Duster we do not see any difference in grain yield, and whereas in NFEX28 we see a biomass enhancement in one field but not in the other. And the same thing here in the next 2 graphs, you see the impact of different cultivars with response to the fungal treatment. So it is the same data with a different perspective. The previous one was grain yield in terms of weight, and this one is grain yield in terms of number of grains. And here also, as you can see those green bars, in cultivar NFEX25 we see an improved grain yield due to the fungal treatment, and in Duster you do not see any grain yield, and whereas in 28 you see a grain yield in one field conditions but not in other field conditions. Lastly, we look at 1,000 grain yield data. This is something commonly the wheat breeders do. And here we do not see any significant impact of these fungi in terms of 1,000 grain yield. So what it means actually, when you use these fungi for improving wheat grain yield, it helps in increasing the grain yield, but it does not change the grain size. So there is no change in the morphology of the grain per se, which is good because we do not want the fungi to change the shape and size of wheat. That's not a desirable trait. So the final take-home message out of this experiment is that when we use these fungi with the help of the delivery mechanism that we have developed, it can increase the grain yield by almost 50%, and this is irrespective of any kind of cultivar which we have tested. However, it does not have any impact on grain size or grain shape. And so basically we have seen that one cultivar, the grain yield was improved in 2 different field conditions. In one cultivar, we do not see any grain improvement, and one is a third cultivar, we see improvement in one field condition but not in the others. So what it actually means, it means that we do not have a single solution for all problems, or we do not have a single solution for all different crops and all different soil conditions. So we have to treat situations case by case. If you see these fungi improving plant health in one condition, that may not be true for a different condition or a different crops. So this is very natural, and we have to consider, we do not have a single rule of thumb that when you use a beneficial fungi, it improves plant health. You have to adapt these good microbes for a particular soil conditions for a particular crops. So with this, I end my talk, and this is our lab. So Kelly Craven is our group leader, and unfortunately we missed him because he has gone for another conference in San Diego. And this is all our lab members. All my research are funded by the US Department of Energy and Nobel Foundation. And with this, I am happy to take questions.